Regulation of the complement system. Um, as I mentioned in one of the earlier clips, one of the main things that regulates the complement cascade system, because you really don't want it to overreact, since it can have negative effects on the human body. If it overreacts, it can do something like stimulate systemic histamine release and cause anaphylactic shock. It could cause actual holes to be punched into normal, healthy cells. If you overwhelm the regulatory mechanisms, that can occur. So you do get a little bit of necrosis from time to time whenever you have major inflammatory response. That's one reason they want to control inflammatory responses in patients. There's a list on table 7.2 that talks about a number of these regulatory mechanisms, and I'm going to try to separate these out into your soluble and your membrane bound. Your soluble components include things like the C1 inhibitor, and it's a protease inhibitor, so it causes the C1R2S2 to disassociate it from the C1Q, which means that it's no longer going to be bound to the surface. Those little golf club heads kind of break free, and it all disassociates from the surface of the cell. This is in fairly low concentration in the cell, but we'll get into that in just a moment. The C4 binding protein. This affects both the classical and the lectin pathways, and it blocks the C3 convertase by binding to C4b. And then, of course, you don't get the C4b binding back. So the cofactor for the cleavage of the C4b by factor 1 is inhibited. Let's see, what are some of the others? The S protein binds to the C5 through C7 complex and prevents it from inserting into the cell membrane. A lot of these proteins are going to be, in the soluble fraction at least, are going to be, one, in relatively low concentrations, and two, they're going to be in, I'm sorry, I got distracted there. They're going to be in relatively low concentrations, and they're going to be more likely to be associated close to the surface of normal, healthy cells. So they're just going to be attracted there, almost like a magnetic pull. So even in a serum solution where they can bounce anywhere, they do tend to congregate around the surface of the cells, especially cells that are releasing them. They don't break away very quickly. And this helps to regulate the complement cascade system and prevent, just random activation of the system anywhere in the human body, or any animal body for that matter. The membrane-bound components, you can have several different kinds of membrane-bound associations on the surface of normal cells, and they have a tendency to block C3 convertase or alternative C3 convertases, like with the lectin pathways. You can have several of them. Let me think. There's the decay accelerating factor, DAF. It's also known as CD55, which just means it's a protein on the surface of most cells, and it can accelerate the C3 convertase breakdown or disassociation, so that unless that C3 convertase is stabilized to a C5 convertase, it has a relatively short half-life. A lot of C3 convertases only last 4 or 5 minutes in normal serum. They think in the body it may even be less time, so they're really small amounts of time. So unless they continue and become stabilized pretty quickly, then they'll break down. So that DAF, or decay accelerating factor, causes the C3 convertase to disassociate. The complement receptor type 1, or membrane cofactor protein, is another of the membrane-bound regulatory mechanisms. It blocks C3 convertases by binding to the C4b or C3b, thus preventing them from being able to stabilize a complex, which means everything just kind of breaks down. And most of these DAFs or MCPs are going to be found on the surface of normal, healthy cells. Let's see. This is interesting. I'm going to actually read part of this paragraph. Complement-mediated lysis. This is on page 179. It's the last paragraph on the page. Complement-mediated lysis of cells is more effective if the complement is from a species different from that of the cells being lysed. This phenomenon depends on a membrane protein that blocks MAC formation. This protein present in many cell types is called a homologous restriction factor, or the membrane inhibitor of reactive lysis, CD59. So you could go on and read the rest of that, but you kind of get the idea that they're wanting to prevent overreaction of the complement cascade, only in an area where the complement cascade keeps being activated through antibodies being bound to an antigen, or reaction to some structure on the bacterial or artificial cell. Only in those areas are you going to get a lot of activation, enough to at least stabilize the C5 through C9 membrane attack complex. That's the end of that clip.